Hello, my name is Nick Verreke and I'm a former PhD candidate from Pathogen and Ghent University. Today I'm going to talk about co-infections in endemic influenza A virus infected herds and how we use nanopore metagenomic sequencing to study the viruses and bacteria present in tracheobronchial swabs. Let's start with a short introduction on the complexity of porcine respiratory disease. Now, if we talk about porcine respiratory disease, we are not only talking about viruses but also about bacteria. Within the viruses, we have some coronaviruses that are important next to Influenza A virus, porcine parainfluenza virus, pseudorabies virus, which is eradicated in Belgium, PCV2. Within the bacteria, we have Bordetella bronchiseptica, Streptococcus suis, Actinobacillus pleuropneumoniae, and Myxomycoplasma hypopneumoniae, which have been described the most commonly. Now, if we think about diagnosing these, some of these viruses and bacteria are quite difficult to diagnose. We have the porcine parainfluenza virus, which was only discovered recently to have an impact in the porcine respiratory disease, so there's very little tests available to date. For Myxomycoplasma hypopneumoniae, it's almost impossible to efficiently grow them, so we have to rely on molecular methods. Now, if we think about diagnostics that are existing at the moment, we can divide them in three different groups. We have the antigen detections, in which we can detect either the immune response or the pathogen itself. They can be quick, but if we check the immune response, we need to wait for at least 3 weeks to make sure we are not looking at the vaccination state. They are cheap, that's why they are most commonly applied. They have a quite low sensitivity and specificity. They cannot detect antibiotics resistance, and there is a semi-quantification available for them. PCR, they are actually the quickest tests that are available to date, however they are also the most expensive. They have the highest sensitivity and specificity. We can do some antibiotic susceptibility testing, however this really depends on which bacteria you're looking at. We have some quantification possibilities since there's qPCRs and there's some assays available for vaccine detection, but they are not that good actually. On the other side, we have the culture-based approaches. We can do viral cultures, but here we will focus on the bacteria as they are normally quite quick, but some bacteria, as I mentioned before for Myxomycoplasma, they are quite time consuming or it is impossible to grow them. They have a quite okay cost, the sensitivity specificity is also okay, and the nice thing here is that we can perform antibiotic susceptibility testing. There is also somehow a quantification possibility attached to them. We cannot detect vaccines here. Now, if we think about the study that we did, this is the methodology. We sampled 25 different forms in Germany, and we sampled 4 different age groups, of which we collected nasal swabs and tracheobronchial swabs, but also we collected metadata on the influenza status and the coughing and sneezing index of all the pigs in this study. Now, all these pigs were further characterized, all the samples were further characterized using the revolutionary technology at Pathogen, which uses nanopore sequencing to look at the metagenomic context of the respiratory disease in pigs. Now, if we look at our results, we could find some viral and bacterial profiles. Here I will divide them in a separate group, the viruses and the bacteria, but just keep in mind that they were obtained from the same data. We will see here for the respiratory viruses that we see a specific pattern for over age. We see that there is no PRRS present in the suckling piglets, which are totally on the left, and we do see an increase over time. The same can be seen for some other viruses, in which you see that the porcine parainfluenza virus in light green is actually present in all ages, and the same is for the PHEV. Now, we can do the same for the bacteria in there, in which we can see clear reduction of the Glycerella and the Streptococcus over time. We also see that the actually 2 bacteria, and that's the Bordetella and the Myxomycoplasma, only appear after the suckling period. Now, what can be done further with this data? We can perform in-depth statistical analysis to compare and look at the beta diversities within these samples. It allows us to actually look at the significant contributors. In this case, we look at age, but we can also do that for the influenza status. We can do that for the health assessment, sneezing, coughing, or any other parameter you're interested in. As you can see here, indeed, for the age, we had PRRS, Myxomycoplasma, Glycerella, Bordetella, and Streptococcus being the respiratory-related bacteria and viruses that were significant contributors over time. That was also what we saw in the graphs I showed before. Now, let's take a step back. This is the metagenomics output, but if we would look at the classical diagnostics, our results would be looking like this. We would not get all the information, but just a subset of information that we actually need to request, because a veterinarian nowadays need to request for each separate bacterium its own PCR, because no multiplex PCRs exist for the porcine respiratory disease. The same accounts for bacteria. bacteria. So if we think we would not get these bar plots, but we would get a report that looks rather like this, in which we get positive or negative, but also some quantification if a qPCR was performed. It can be for viruses and bacteria. For bacteriology, we should also do the bacterial cultures, and we would get the MALDI-TOF MS identification of the bacteria. The nice thing here is that we could get the antibiotic susceptibility testing. Now, as you can see, there's some exclamation marks for this PRRS virus, which is a very important one, because here we also need to consider the vaccination strategy. Because pigs get vaccinated with live attenuated vaccines, so they are also actively replicating. So the question is, if a high positive PRRS sample, does this mean we have the wild type, or does it mean we have the live and attenuated vaccine that we detect? That's why we have to perform the classical method, the classical molecular diagnostics, and we have to supplement this with sequencing analysis. This would give us allow us to look at the phylogenetic inference of this virus and classify them as the blue wild-type vaccine, wild-type strain, or the red one that is actually the live attenuated vaccine. So you can conclude if you have a wild-type or a wild-type infection, or if you're detecting the vaccine in your strain. These are quite expensive if you have to request for these analysis. Now, the conclusions and perspective of this work, as you can see here, this is the comparative table as I showed before. The metagenomics has quite good in its time. It has a high cost, which is in the level of multiplex PCRs. Its sensitivity and specificity is good, plus it can also identify all Mycoplasmata C, which are almost impossible to grow in many cases. It is also not dependent on the host species. We can perform antibiotic susceptibility testing, however, this is work in progress. For who has attended London Calling in the past, you might have seen my previous presentations on how we try to tackle this problem. We also have a semi-quantitative feature to this metagenomics, as we can obtain relative abundances, and we also have the vaccine that can be resolved because we get the complete viral genomic sequences in many cases. We can differentiate the vaccine, and we can also resolve potential recombination. Now, in conclusion, the metagenomics can identify viruses and bacteria plus the viral subtyping. We can have the complete community statistics as we do the beta diversity for age dependency in the community, but we can also assess important players in swine influenza positivity and symptoms like sneezing and coughing. We can identify lesser studied pathogens for which there's actually no data available, or we have no idea they are still circulating, which was the case for PHEV and percent para-influenza virus. You want to know more about this research? All this data got recently published in Microbiology Spectrum. Just scan the QR code and you can read our paper there. Do not hesitate to follow us and contact us with any further details. I still want to thank all the collaborators, because without them we would not have been able to do this work. Thank you for listening.